rewatch me talk subjects and stuff on your own time, you are more than welcome to. So the first thing that I wanted to show you guys, let me pull it up here. All right. So just as a reminder, I briefly mentioned it last week, but the kind of big thing going on this week, besides all of our normal notes and assignments and stuff, is that on Wednesday, our first midterm exam, our first test in this class, is going to be posted. So it'll go up tomorrow morning, so right as the day starts, 12:01 a.m. But it is not due until Friday, November 20th. So you can see I wrote it in the title that it's due Friday, and it says even down here, due November 20th. So you have 3 days to work on the exam. You have all day Wednesday, all day Thursday, and all day Friday. I wanted to give you some extra time to work on it because it will be longer than your kind of normal daily homeworks and assignments and stuff like that. So I think it's about 24, 25 questions, somewhere around there. And it covers all of the stuff that we've talked about so far in class, from the DNA to the replication to the protein synthesis with the mRNA and the genetic code with the codons and the AUGs and CCCs and GAGs and stuff like that, as well as some of the stuff that we're going to talk about this week with cell division, the mitosis and meiosis stuff. That will be on the exam as well. And so it's a mixture of different types of questions. So there'll be some multiple choice questions, which on the Google Doc version, you can just highlight or bold or write in the letter of the answer for your multiple choice question. There'll be some matching questions with the DNA. There'll be some short answer questions where you have to write down the answer in your own words. And usually like 1 to 2 sentences is about as long as you should need. There are some fill in the blank questions. There's some labeling questions. So all sorts of different types of questions. Again, covering everything that we have talked about since the beginning of the class. That's what the Google Doc version looks like. If you want to do the Google Form version, which I know a lot of people like to do, it has the exact same questions in the exact same order as the Google Doc version. It's just spaced out a little bit differently. So the questions are divided up into sections. So the first section is just sort of this introductory section where I have you write your name to make sure I know who turned in what. And then there's a DNA replication section with some questions in here. And then after you fill out all those questions, you'll come down here and hit next, and it'll take you to the next section. One thing to note about the Google Form version is that it will not save your answers until you hit the submit button. So if you're working on the Google Form version and you accidentally close the window or you close the tab or something happens with your internet, then your answers might not get saved and you may have to redo some of the questions. On the Google Doc version, it'll automatically save any questions that you answer. So it'll automatically save any changes that you make to the document. So again, that is the midterm It'll be posted tomorrow morning, so I have it scheduled to go up tomorrow morning. And then it'll just due on Friday the 20th. So at the very end of the day on Friday. Again, it's not like a huge exam. It's not like 100 questions or anything crazy like that. But it still does cover a lot of information. So I wanted to make sure to give you guys plenty of time to work on it as much as you need over the course of the week. And this way, once you get finished with the exam on Friday, you get a whole week off of school to rest and relax over Thanksgiving before we come back the week after. Obviously, since it's an online exam, I don't have any restrictions on going back and looking at the notes or looking at the class videos of stuff that we've talked about. I would recommend against Googling for answers, just because sometimes the answer that Google brings up isn't exactly the same answer that we talked about in class that you might have been looking for. And also, sometimes it's really easy for me to tell if you just copy an answer from Google. Like if you give a super technical, precise answer on your midterm, I'll know that it probably wasn't you. So make sure that you're using your own words when you write in your answers, and use the Google Classroom to your advantage. So go back, scroll down a little bit, and look back at some of the notes from previous classes to help you out. You can also reach out to me if you ever have any questions about the exam. Obviously, I can't help you directly answer a question, like I can't help give you the answer. But if you're confused about what exactly the question is asking, or you're not quite sure what type of answer you're supposed to be giving, that is something that I can help you out with. So please reach out to me if you get stuck on one point and you're just not quite sure what to do. All right. Any questions related to the midterm coming up this week? All righty then. If not, then we will go ahead and jump into the notes for tonight. So just like last week with the protein synthesis notes, it's a similar situation this week where we have one set of notes that's really big. So we're going to be covering it over the course of both tonight and Thursday night. And these notes are specifically about cell division. So how do we take our cells, grow them, and split them into new cells? All right. So cell division, it's the process in your body by which we make new cells from your existing cells. So you may have noticed over the past 20 to 30 years of your life that there is more of you now than there was when you were a kid or a toddler or even a baby, right? No matter who you are, how big you are, there's a lot more of you now than there was 20, 30, 40 years ago. And that's not just because you put on weight out of nowhere, it just appears on your body. It all happens due to cell division, cells growing bigger and then splitting into two new cells. So today we're going to talk a lot about kind of the process behind how cells get ready for this division, as well as kind of the basic plan for cell division, which is known as mitosis. So the first thing that we have to talk about when we start talking about cell division is we're going back to our DNA. Ooh, hold on, where my little, there we go. our DNA. So we're still talking about DNA. That's how important it was. So in our cells, DNA doesn't just hang out as a big double strand all the time. Instead, it gets wrapped up and wrapped up and wrapped up and condensed into something called a chromosome. So a chromosome is DNA. It is made out of DNA. It's made out of DNA, but the DNA has just been condensed down into a smaller package, right? There is a lot of DNA inside your nucleus, and it's not all going to fit if it's just kind of all hanging out loosey-goosey. So instead, your DNA gets squished and condensed and wrapped around itself into structures called chromosomes. So when we were talking about DNA, you may remember that I mentioned that every single living thing on Earth has DNA inside of its cells. We're all based on DNA. Whether or not you're a spider or a mouse or a human or an elephant, everything is built out of DNA. So all of your DNA is called your genome. So all of the bases that make up your entire DNA sequence is called your genome. And like we've been talking about, your DNA has the instructions for your cell, specifically for your proteins, right? Last week we talked about how each gene has a code in it, and that code is for a specific protein. So if you were to stretch all of your DNA out when it was just in its double-stranded form, if we stretched it all out end to end, it would stretch about 3 meters long. So that is a lot of DNA. And remember, every single one of the billions and billions of cells in your body has a complete set of DNA inside of it. So if we just let it be all stretched out, it ain't gonna fit. Instead, it gets packaged down into these structures called chromosomes. So a chromosome is essentially a condensed or packaged form of DNA. So we have lots and lots of DNA, and we compress our DNA down into structures called chromosomes. So your DNA is not just one big long strand or one big chromosome in your body. There's actually a lot of segments of DNA. And so when we condense these segments down into chromosomes, we end up with, in the case of humans, 23 different DNA segments, or 23 different chromosomes. The interesting thing about your DNA in your chromosomes is that you do not just have one copy of your DNA inside every single cell. Instead, you have 2 copies. So every single piece of DNA, every single chromosome in your cell, in your nucleus, occurs in 2 copies, a pair of chromosomes. And we call these chromosomes homologous chromosomes. So these are our chromosome pairs that exist in most sexually reproducing species. So in any sort of animal or plant where you have to have a male part and a female part come together to make a baby, you have homologous chromosomes. So copies of every single chromosome. The reason why you have 2 copies of every single pair of chromosomes is that you get one chromosome from your mom and one chromosome from your dad. So the reason why we have all of these pairs of chromosomes is that one copy came from mom and one copy came from dad. So these copies are identical. So every single copy of your DNA has the same genes on them in the same order, same everything. The only one that is changed is your sex chromosomes, your X and Y chromosomes. So you always inherit one X chromosome from your mom. And then from your dad, you either inherit a Y chromosome or an X chromosome. If you get a Y chromosome from your dad, then do you know what the gender of the baby will be if they get a Y chromosome? Does anybody know?
I'm going to take a wild guess. A boy?
A boy. That is correct. So in the sex chromosomes in humans, X and Y means that you get a boy.
Boy, yes.
Whereas an X chromosome from your mom and an X chromosome from your dad means it'll be a
girl, yes.
So those are the only chromosomes that are different. All of the other chromosomes, all of the other sets, all the other pairs are all the same. They're all identical. It's just one copy comes from mom and one copy comes from dad. The XY is the only one where you get different copies from your dad than you do from your mom, potentially.
Excuse me, teacher?
Yes.
That's mean Y and X equal girl?
A boy.
A boy, okay. Y
so
and
XY is boy. XX is girl.
X is girl, okay.
Precise. So each one of your cells has DNA inside of it. This DNA is condensed down into sections called chromosomes that are really condensed, packed in DNA. You have two complete sets of chromosomes in each cell, one set from your mom, one set from your dad, and we call these sets homologous chromosomes. So the pairs, so here's one set of homologous chromosomes, here is another set, here is another set, so on and so forth. So in each set of chromosomes where you get the homologous chromosomes, so one copy from your mom, one copy from your dad, they have genes that code for the same traits. So we've talked about genes already with our DNA. Genes are those segments that contain the code for a specific protein. Remember from last week, our gene gets copied into an mRNA sequence, and then that mRNA goes to the ribosome and gets turned into a protein. So each homologous chromosome contains the same sets of genes. So here is a actual picture of some genes that some scientists took under very high-powered microscope over a long period of time, where they were able to separate out the different chromosomes. And so again, each set of chromosomes has different genes on it. So like the genes on chromosome number 1 are different from the genes on 2, which are different from the genes on 3. But each homologous chromosome in a set contains the same genes on it. So these guys both have copies of the same gene. These guys both have copies of the same genes. These guys both have copies of the same genes. And on and on and on and on and on, with the only differences being the sex chromosomes, where you could have an XY for a boy or an XX for a girl. But those are the only chromosomes that are potentially different. All the other ones are the same as each other. So I started this presentation saying that I would tell you guys about cell division, and I promise we're getting there. So when a cell is getting ready to divide, when it's grown and it's gotten to the point where it's ready to start splitting to become two different cells, each of those new cells has to have a copy of DNA. And so before the cells split, we have to duplicate or replicate our DNA. And remember, this is what we talked about during that second week of class. So when we talked about the polymerases putting down the bases and the 3' and 5' sides and the primers and the Okazaki fragments, all of that was due to DNA copying itself because the cell is getting ready to divide. And if each of our new cells is going to have a set of DNA in it, then we have to copy that set inside of the original cell. So if we start off with 2 copies of each gene, one from your mom and one from your dad, and then we make duplicates of all of our chromosomes, all of our DNA, then we end up with 4 total copies after we duplicate it, right? We started with 2, and then each one of those got replicated. So now we have 2 copies of your mom chromosomes and 2 copies of your dad chromosomes. And the 2 copies together are called chromatids. So I know this is a lot of terminology that I'm throwing at you guys. I'm trying to take it a little bit slow here at the beginning. So again, homologous chromosomes are that pair where you get one from your mom and one from your dad. Chromatids are what you get when that pair duplicates. So the 2 copies that you now have from your mom are chromatids together, and the 2 copies of your dad's chromosome are also called chromatids together. I just bring it up because you may hear me refer to them as chromatids or sister chromatids later on in the notes. And so I just wanted to give you guys a heads up of where that's coming from. All right, so final thing with chromosomes is chromosome number. How many segments of DNA are in each cell? So it actually is very different for each species. So not all organisms have the same setup with their chromosomes. And also, having more chromosomes or having more DNA doesn't necessarily mean that you're a more advanced or a more complex species. So in a somatic cell, so somatic just means like a normal, average, everyday body cell. So like skin or muscle or heart or stomach or eye, all of these are somatic cells. and they contain what is called the diploid number of chromosomes. We'll talk about diploid and haploid more in a second, but diploid is essentially the kind of the average, the normal number. So like a diploid cell has those two copies, those homologous chromosomes, the one mom copy and the one dad copy. Most cells have those two copies in them. And so they're called diploid cells because they have two copies of every chromosome. Specific cells known as gametes, which are also called sex cells. So in males, we have sperm cells, and in females, we have egg cells. These have half the normal number of chromosomes, so they don't have two copies. They just have the one single copy. And this is what's known as the haploid number. So haploid, single copy, diploid, two copies. Again, most of your body cells, 99% of the cells in your body are somatic cells, and they have two copies in them. Only the sperm or egg cells are the gametes and have the single haploid copy. So diploid, normal double copy, haploid, single copy. So species are all over the place in terms of how many chromosomes that they have. So camels have a total of 70 chromosomes. Again, this is the diploid number. So this is if we count all of the copies, how many do they have? Squirrels have about 40. Chickens have 78. King crabs have a ton of chromosomes, 208 total. Fruit flies on the other side of the spectrum, they only have 8 chromosomes. Tomatoes have about 24 total chromosomes, and potatoes have about 48. So again, even though tomatoes and potatoes have way more chromosomes than fruit flies, this doesn't mean that they're more advanced or have more genes. It just means that they have longer stretches of DNA. Humans fall kind of right in the middle of our spectrum here. We have 46 total chromosomes in our cells. So 46 total chromosomes because we have 23 pairs, 23 different pairs of chromosomes. So you inherit 23 chromosomes from your mom, and you inherit 23 chromosomes from your dad, put them together, and we have 46 total chromosomes. So again, in our haploid cells, in our half number cells, like our sperm and our eggs, there's only 23 different chromosomes, or 23 chromosomes in there, one copy of each of the 23. The 99% of cells are normal diploid cells. They have two copies of the different 23 chromosomes. So we have 46 chromosomes in a normal human cell. 23 that we inherited from our mom, and 23 that we inherited from our dad. So there are 23 different chromosomes in the human body. In haploid cells, we only have one copy of each one of those different chromosomes. So we have 23. In our diploid cells, we have two copies of every one of those chromosomes. And so there's 46 total. Does anybody have any? So we'll get to that in a second. I know there's a lot of stuff that I just threw at you about chromosomes and chromatids and all sorts of fun stuff like that. Does anybody have any questions so far on what chromosomes are, why we have them, or how many we have?
No.
No.
All righty. Having exactly 46 chromosomes in our body is actually extremely important for normal growth and development. So you have to have all 46 chromosomes to function properly, quote unquote. So even being off by one chromosome, missing one chromosome from your dad, or getting one too many chromosomes from your mom, can have extreme serious implications for your health and development. So a lot of times, infants that inherit the wrong chromosome numbers won't even start developing properly. I shouldn't say infants, the zygotes, the fertilized eggs, will not start developing properly and actually cause a miscarriage in the woman, even if there's only one chromosome out of place. So monosomy means you only have one copy of a chromosome you're supposed to have two of. Trisomy, having an extra one, means you have three copies of a chromosome, even when you're only supposed to have two. Actually, hold on, let me see if it. So actually, we'll get back to trisomy in a second. So the way this happens, just really quickly, is that normally the chromosomes are supposed to separate and split equally when the cell is dividing. But sometimes that doesn't happen, and we get too many chromosomes pulled one way and not enough chromosomes pulled the other way. And so we end up with a cell that either has one too many chromosomes or one too few. It doesn't have the correct number. So here is another picture of the chromosomes inside of a human. Again, you can see we have 22 normal pairs plus a sex chromosome pair over here. I think that's two X's. Does anybody see what is wrong with this particular set of chromosomes?
21 has 3.
Desiree has spotted it. 21 has 3 chromosomes instead of 2. And so in a medical setting, that would be called trisomy 21, because chromosome 21 has 3 copies, which is unusual. Does anybody know the relatively well-known developmental disorder that is caused by having 3 copies of chromosome 21?
Down syndrome?
Down syndrome. Noberto got it precisely. So you've probably seen somebody with Down syndrome before. They have particular facial features and their learning ability is severely hampered. It takes them a lot longer to learn things. So their development, their mental development is affected. And all of these properties come from simply having one too many chromosomes in the number 21 spot. So just by getting a third chromosome, just by inheriting a third chromosome from one of their parents, causes the child to develop all of these developmental disabilities. So having exactly 46 chromosomes in your normal body cells is extremely important and is something that your body tries really, really hard to make sure happens. All right, so we're getting this DNA copied. We're having all these chromosomes be bunched up and in our cells. And it's all because we're getting ready to divide. We're getting ready to go through cell division. So like I was saying, cell division is the process by where we take one cell and we split it so that it becomes two cells. So obviously there is a lot of sort of behind the scenes stuff in order to make this happen that your body does. It's not just we take a cell and just slice it in half and we end up with two new cells. That is not it at all. We need to make sure that everything in the cell is copied and ready to go before we split into two parts. So cells divide all the time. There are cells dividing in your body right at this moment as we speak. And they do it for all sorts of reasons. Maybe you need to grow if you're a child growing into an adult. Maybe you need to repair some damaged tissue. Maybe there are cells that die, which happens. There are constantly cells in your body that are dying and need to be replaced. All of this requires cell division in order to create new cells. We also have to go through cell division in order to create our sperm and egg cells that'll eventually join up together to start creating a new offspring. So when you're very, very first conceived, you start off in your mom as one single cell. Then that grows and splits to become two cells, which grow and split to become 4, and then 8, and then 16, and then 32, and so on and so forth. So all of these cells in your body right now are all directly descended from that one single cell that started off inside of your mom. So as cells grow and get ready to divide and grow again, they go through something called the cell cycle. So just like humans and dogs and cats and birds have life cycles, cells have cell cycles. So it's like the life cycle of a cell from when it's first created to when it splits to create new cells. So there are 5 cell cycle stages. So right after the cell splits, so right when we've got a fresh, brand new cell, we go into stage G1, which is the first kind of initial growth phase of the cell. So it just starts to grow and develop into its role that it's playing in the body. Eventually, after G1 has gone on for a while, the cell will get a signal that says, hey, it's time for you to divide once again. When it gets that signal, it'll then go into S phase. And you may remember hearing about S phase before. We talked about it during week 2 of class, because the S phase is when we copy the DNA. So all of that DNA replication stuff that we talked about in the second week with the leading strand and lagging strand and the helicase and all that good stuff, that all happens during S phase. So we know that we're gonna divide the cell at some point into 2 new cells. So we've got to copy all of our DNA to make sure that it gets passed on to the new cell. That happens in S phase. G2 is the next stage. This is kind of where the cell starts growing again, but it's growing because it's getting ready to divide. So we'll talk about it more in a second, but the actual process of cell division normally is called mitosis. So we're getting ready for mitosis, for splitting our cell, and we're copying all of the little structures inside of the cell, which are called organelles. So these first 3 stages, the G1, S, and G2, are known as interphase. And that's because that besides kind of this S phase when we're copying the DNA, this is all just sort of an in-between phase between one round of division and another. So because it's the in-between phase, scientists called it interphase. We then move into M phase, where we actually go through the process of splitting the cell apart into new cells. So M is for mitosis, for cell division, where we actually split the cell into 2 new ones. And then finally, the final stage is referred to as C. It stands for the name of cytokinesis, which essentially just means that the cell finishes kind of pinching off from each other. So during the M phase, the DNA lines up and splits apart to opposite sides of the cell, which we'll talk about more in a little bit. And then the cells start to split apart from each other. But the C phase, the cytokinesis phase, is where they finally officially split. So that's like they're separated, they're gonna go their separate ways, except not really because they're cells, so they're probably jam-packed in right next to each other. But C is like the final split, the final cut that causes them to separate and become 2 fully formed new cells. So G1 and G2 are sort of our in-between growth phases. S is where we copy the DNA. And then M is where we split the cells apart. And C is where we do the final cut to make them into 2 different cells. So I like this diagram because it kind of shows you everything going on and approximately how long that it takes. So those final 2 steps, the mitosis and cytokinesis, are very, very short in terms of the life cycle of the cell. They happen pretty fast. The other stuff is really where most of the cell's time is taken up. So it spends most of its life in the G1 stage. Remember, that's just it growing and developing and doing all of its normal cell stuff. And then S is when we start to get ready to divide. And so we copy our DNA. So the S is for synthesis, which is just a fancy science word for making or building stuff. So we're making DNA in S phase. And then we do some more growing and getting ready to divide. Again, all of this is known as interphase. So G1, S, and G2 are all together as interphase. And we spend all this time growing, and then we copy the DNA, and we have a lot of DNA, so that takes a long time. And then we do some more growing, and then finally it's like, and boom, split everything apart, boom, cut the cell in half. All right, now we got 2 new cells. And now we're gonna go through that whole process over again. So growth, copy the DNA, some more growth, split the cell apart, cut it apart, new cells. And that is sort of a general overview of what's going on during the cell cycle. It's been a while since I asked, so any questions so far about the cell cycle or about anything that I've talked about with the chromosomes or the cell division here so far tonight?
No.
Awesome. Thanks, Paula. Anybody else? Any other questions? All right.
I'm good.
All right. Thanks, Namerta. So there are a couple different things that can signal to a cell that it is time to divide. One of them is size. So for a lot of cells, it's just once they hit a certain size, then they get the signal like, hey, it's time to divide. You're getting kind of too big on the inside to be contained within your outside. So dividing helps it to kind of separate out its important parts, but then cut itself in half and kind of restart over from a smaller size so it can start growing once again. If there's certain areas of your body where you're growing, especially when you're younger or you're going through puberty. So growth factors are chemicals that cells release that can encourage cells nearby to divide. So they signal to other cells nearby, like, hey, we got to keep this momentum up. We got to keep growing. We've got to put 4 inches on this kid by the end of the summer so that he looks really weird when he goes back to school next year. Keep on dividing, keep growing, keep dividing. Let's keep it up. So those ways can kind of cause cells to divide. Usually it's size. So usually it's just a cell is getting too big. And instead of just stopping its growth and sitting there, it will split in half. But sometimes it's due to these kind of external triggers as well. All right, so when our cell is actually gonna go through that meiosis, that M phase, and split, there are actually not just one, but 2 types of cell division that a cell can go through. So the first one, and the one that we'll finish off talking about tonight, is called mitosis. So in mitosis, we take one cell, we split it apart, and we end up with identical cells. So the new cells that we get are exact copies of the cell we started with. So mitosis is what happens in your normal body cells. So those somatic cells that I was talking about earlier, that's where mitosis happens. And at the end of mitosis, we end up with diploid cells. Again, this is the normal state for the cell to be in. So we basically just make 2 Xerox copies of the original cell. So we start off with our original cell that again has 1 copy of each chromosome. So this is showing like, oh, here's 1 chromosome. You got the red one from your mom and the white one from your dad. We copy those chromosomes to make those chromatids, since we duplicate the DNA. And then when we go through mitosis, we split that DNA apart and we end up with 2 new cells that are identical to our original cell, right? Starting cell, ending cells look exactly the same. So mitosis is just regular cell division, regular cell growth. This is what 99% of the cells in your body go through. We start off with a normal body cell. It grows and splits, and we end up with 2 identical copies of that original cell. That is mitosis. So I always try to remember it that mitosis makes 2 cells. So there's a 2 in mitosis because we make 2 cells. So again, we start off with 1 cell and it has a whole bunch of DNA on it. And then we copy that DNA, we split it apart from each other. It goes to either side of the cell and eventually causes a split up to become 2 new cells. We'll go ahead and skip this for now. Usually if we were in class, I'd be asking people like what order do they think all the different steps are in for the different pictures. But we'll skip this for now. We'll actually come back to this and kind of talk about what the actual order is after we go through mitosis. All right, so mitosis has a series of 4 steps that it goes through in order to make sure that all the DNA gets separated to each new cell. Because that's kind of like the main thing that the cell is worried about, is that the new cells get the correct amount of DNA. So we go through 4 steps in mitosis to make sure that our DNA gets separated the exact same way every single time. So the first step of mitosis is called prophase. So in prophase, the chromosomes actually become visible. So before, they were kind of hard to see inside of the cell. They were inside the nucleus, and the DNA wasn't quite as tightly packed as it could be. But in prophase, those chromosomes get really packed down and really dense, so it's easier to see them. And also, the nucleus breaks down, right? So we've talked about how the DNA is inside of the nucleus in order to be protected. That's why when we're making proteins, we have to have that mRNA strand that kind of goes back and forth, that comes out of the nucleus to the ribosome, because the nucleus is surrounding our DNA. Well, in order for the cell to split apart, that nucleus has got to go. So the nucleus breaks down and the DNA really condenses tightly into the chromosomes, which makes them easier to see. We also get some new structures called asters and spindle fibers that appear. So we'll talk a lot more about the asters and spindle fibers in the later segments, but essentially these are the little structures that are going to be responsible for pulling this DNA apart, for separating it into what is going to become the 2 new cells. So keep your eye on these asters and these spindle fibers, because they're going to be important. So this is prophase. This is kind of like the first setup phase of mitosis. We're getting things ready. That nucleus is going away. The chromosomes are getting really tight and condensed. And we've got our first little fibers starting to come out. So eventually, we're going to need to separate these chromosomes, right? Pull apart all those copies so that each new cell can get the right amount. And the spindle fibers are what are going to be doing the pulling. So here is an actual picture of a cell in prophase, kind of the very end of prophase. You can see the chromosomes are super dark and condensed. There is no nucleus anymore, so usually there'd be like a circular nucleus covering this. That's not there. The asters, you can't really see them, but they're like the little nodes on either side of the chromosomes here. And then all these little webs, all these little strings going back and forth, those are the spindle fibers. So the nucleus is gone, and the spindle fibers are starting to reach out towards the chromosomes. So that is prophase, kind of the first setup phase of mitosis. The next setup phase is called metaphase. So metaphase is always really easy for me to remember because meta means middle or center. And so in metaphase, and also it's M for middle. So metaphase, the chromosomes light up right up and down the equator of the cell. So like right up and down the middle of the cell is how the chromosomes get positioned. So before, those chromosomes were all kind of really condensed up and they're all just kind of floating around where the nucleus used to be. In metaphase, the little spindle fibers attach and cause them all to kind of line up right down the middle of the cell. So whenever you see a cell, kind of like this little one down here, where we've got all these guys lined up, or this one right here is especially good, you can see that we've got all of our chromosomes lined up right in the middle of the cell and all the little spindle fibers attaching to them on either side. So again, metaphase is the middle phase. This is where we all get lined up. We all get ready. So prophase is, what's a good way to remember? Prophase is like the primary phase. So the very beginning, stuff is breaking down and forming and kind of getting set up. Metaphase, the middle phase. So all of our chromosomes are now lining up right down kind of the middle part of the cell. All the spindle fibers are coming in and attaching and hooking on to the chromosomes. And that's because up next, we have anaphase, which I like to call it the action phase. This is where the actual split of the DNA occurs. So in anaphase, all those chromosomes that were lined up along the middle of the cell get pulled apart, get separated out. So those duplicates, those chromatids that were duplicated, they get pulled apart from one another. So that this way, each new cell gets an exact copy of all of your DNA. So each chromosome is getting pulled by the spindle fibers and is going to the opposite ends of the cell, kind of the opposite poles of the cell. So all of our little fibers in here are pulling on these chromosomes and causing them to move to each side of the cell. They want them to be far away as possible so that when the new cells start to form, there isn't any overlap. We have one full set of chromosomes over here, one full set of chromosomes over here. All right, so metaphase, the middle phase where they all get lined up, leads directly into anaphase, the action phase, where they get pulled and split apart by those little fibers. Finally, Okay, are you guys there? Can you hear me? Do you see me?
Yes, teacher.
Yes.
Oh, okay. The
Yes.
internet here at school went out all of a sudden.
Mm-hmm.
And so I'm trying to what was that? Okay, sorry. Let me see here. Can I share from my phone, maybe? All right. All right, here. Give me 1 minute to try something on my computer, and if not, then we'll figure it out phone style. All right. Let's try this. Well, luckily we were really close to the end of the notes. Yeah, okay. So it looks like my computer network is down here. But we can. This is going to be interesting. All right, so we're going to try doing this from my phone. Can people see the Google Classroom screen right now?
Yes.
Oh, okay, interesting.
Mm-hmm.
Okay, Give it a second to load. All right, so can you guys see the cell division slides?
Yes. Cell division.
Yes.
Hot dang, that's
Okay.
pretty sweet. All right, well, we might have to end this class a little bit differently than normal. Luckily, we were right at the end of where I wanted to get to today, which is good. Let me double. Yep, does not look like the school internet is back. All right. So we ended talking about telophase, which I'll put kind of right here in the middle-ish. So telophase is our final stage of mitosis. This is where, now that everything is fully separated, the chromosomes don't have to quite be condensed anymore, so they start to become a little bit looser. And we get a new nucleus forming around each of them. So each new cell now has a nucleus starting to form back up there to protect the DNA. So now we've totally separated our DNA. We have one set of DNA on one side, one set of DNA on the other side, and they're starting to form into two new cells. We're starting to get the nucleus forming back up, and everything has fully been put on either end. So after telophase, the last step that happens right after mitosis is this weird word, cytokinesis, which just means that the cell membrane does its final kind of split and you end up with new cells. So like before, when we were looking at telophase up here, you'll notice that they're separate, but not quite split apart yet. We still have a little tiny bit connecting the two cells to each other. And cytokinesis, that's where each cell develops its own membrane, so its own covering around the outside. And so the cells fully separate from each other. So when you see cytokinesis, I want you to think separate. We snip apart the cells and end up with two brand new identical cells. So in mitosis, we end up with two identical, what are called daughter cells. So in this cell division, we call the new cells daughter cells and the original cell the parent cell. So each daughter cell is identical to the other one. It's like identical twins. It's also identical to the parent cell. So we just made an exact Xerox copy of the parent cell, and now we have two cells that are the exact same. So we had our original parent cell. In prophase, the nucleus disappears, the chromosomes start to bunch up and get thicker, and the little fibers start to form. In metaphase, everything lines up right down the middle of the cells. The fibers hook on and attach. Because in anaphase, the cells will fully separate apart from each other. They'll pull that DNA apart onto either side of the cell. And then telophase, the spindles kind of disappear and the new nucleus starts to form on either side. Ooh, it looks like maybe my school internet is back. Hold on, let me try and get my computer hooked up. Ooh, it looks like it is. Okay, hold on. We might be able to go back to normal mode. Okay, I'm back. Do you guys hear me? Do you guys see me?
Yes.
Hey.
Uh-huh.
All right. Sorry about that.
It's okay.
Hopefully, I was still able to get everything across all right. Okay. This we already went through, splitting the cells apart. So again, mitosis, prophase, nucleus disappears, chromosomes and spindles start to form. Metaphase, everything lines up right down the middle. Anaphase is the action phase, so we split apart our DNA onto opposite sides. And then in telophase, the new cells start to separate from one another and a new nucleus forms. And then it's not in this picture, but in our final step, our cytokinesis, we're just going to cut these cells apart from each other. So here you can see different cells, and this is actually, I think, onion cells zoomed way in close up that are in different stages of mitosis. So you can see this guy over here is probably in prophase, some part of prophase, because the chromosomes are really visible and the nucleus is gone. This one I would say, is probably metaphase, or at least getting close to metaphase. We've got our chromosomes that are starting to kind of line up down the middle. And then over here, we have our anaphase, our action phase, where we're splitting the chromosomes apart from each other and pulling them to opposite sides of the cell. My telophase is kind of hard to picture. I can't really see an exact picture. Maybe these two guys right here, where they're kind of separate, but it looks like the nucleus might still be forming a little bit with these ones. But regardless, those are the steps of mitosis that the cell goes through in order to make sure that we get the correct amount of DNA on either side of the cell. So here's kind of a little bit cooler image of it. So here's our normal cell and interphase. So remember the in-between phase, we're just kind of chilling, getting ready to divide. And then when division starts to happen, have prophase, where the nucleus isn't there so much anymore, and you could really see the chromosomes starting to fill in. Here's a little bit later, where the nucleus is completely gone, and we've got those spindle fibers really starting to form. Metaphase, all of our blue chromosomes are kind of lining up right in the middle of the cell here, and spindles are starting to hook in to get ready to pull. Anaphase, we have the action phase. We pull apart the chromosomes, we separate them to either end. And then telophase is where we start to reform that nucleus, and we start the process of fully separating the cells apart from each other. And then again, cytokinesis will be when the cells fully separate and become two new identical daughter cells. So again, we go through this whole process all so that we can end up with two diploid cells. So they each have two copies of each chromosome. So two blue copies and two red copies. And that are identical to the initial cell. So both of these cells should have the exact same DNA, the exact same structures, the exact same proteins as that original cell. Any final questions about cell division, mitosis, the different stages? I know we got a little bit kind of discombobulated in the middle right there. But does anybody have any final questions about any of the different stuff that we talked about with the cell division or mitosis so far this evening? All right. Well, I think we've come to a good stopping point there. Next week, we'll talk about a different form of cell division known as meiosis. That is a little bit more complex than mitosis. It has a couple more steps and stages involved. And so we'll talk about that more on Thursday. Thank you guys so much for hanging with me in there despite all of our various technical hiccups. As always, if you do have any questions about anything that we've covered in class tonight or anything that we've gone over this term at all, you can always text me, email me, leave a comment on the Google Classroom, and I'll get back to you as soon as I am able to. Also, always go back and look through the notes that are posted up on the Google Classroom or at the video that I'll be posting of this meeting here and shortly after we finish up. Again, remember that the midterm is going up this week on Wednesday, which is tomorrow. So make sure you check that out and work on it some. You don't have to finish it all in one day, but make sure Wednesday, Thursday, and Friday, you're working on it and getting it finished. Oh, one last thing I wanted to go over really quick. I almost forgot. Even though the midterm is coming up, it does not count as one of your daily assignments. So there'll still be an assignment for Monday, Tuesday, Wednesday, and Thursday of this week. So we did the Monday assignment yesterday. It was a fairly quick one to do with creating some proteins. What am I trying to do? That's right. There we go. So the assignment for tonight is some basic questions about chromosomes and the cell cycle. So like the number of chromosomes, what are chromosomes, what happens in different stages of the cell cycle. And then the assignment going up on Wednesday has to do with different phases of mitosis. So it has a whole bunch of kind of notes at the beginning that kind of review these different phases and what happens in them during mitosis. And then has a series of questions at the end that are partially based on the notes we went over, but mostly based on these notes right here. So it should be pretty straightforward to be able to figure out the answers to these questions based on the notes in the worksheet. All right, so make sure you do that assignment tomorrow so that you're able to earn your Wednesday attendance credit. And don't forget about the midterm, which goes up tomorrow, but is not due until Friday. So you have all day Wednesday, all day Thursday, and all day Friday to work on the midterm. All right, any final questions about any of that before we log off here tonight? All righty, guys. Well, thank you so much for being here. Thank you so much for sticking with me through the technical fun times. Please reach out to me if you have any questions about anything. Have a good rest of your evening,